Okay, today we're going to start talking about genome-wide approaches, starting with next-gen sequencing, but going on to a few more applications. Okay, so next-gen sequencing is also called massively parallel sequencing, and it has a lot of similarities to the good old Sanger method that we're familiar with, but the outcomes are hugely different, and there's some major differences in how it's actually performed. Okay, so literally millions of base pairs of DNA are going to be sequenced all at the same time. Okay, so we're just going to do the basic version, but there are many, many applications of this type of technology, many variants. Okay, so looking ahead, we have many steps. The first is library preparation, attachment of DNA to a flow cell, PCR amplification on the flow cell to form clusters of DNA, which will be sequenced. Then there will be sequencing by synthesis using fluorescent chain terminators that are actually reversible, and that's kind of key in this. Following that, we will get alignment of our reads to annotated genomes and very complicated DNA analysis. Okay, so here's a picture of a flow cell. It's a glass side, and it has oligonucleotides attached to it. So that's where the DNA will be bound and where the sequencing will occur. Okay, so there's 8 lanes on a typical flow cell, but you can actually load more samples than that using specific changes in your DNA adapters, but we won't get into that. Okay, so there's oligonucleotides attached to the flow cell. And the idea is that these are going to capture the DNA that will be sequenced and that we will be able to then amplify in place. And so we will generate clusters of identical molecules that will be sequenced all together. And this is going to be happening in millions and millions of spots all along the flow cell. Okay, so the first step, this is for genomic DNA, and that's fine. We have to prepare our sequencing library. And so first we obtain genomic DNA, of course, fragment it into small pieces, and then crucially ligate adapters to the ends of the molecules for the purpose of, first of all, sticking them to the flow cell, but also for the purpose of amplification and sequencing. So the adapters are important for this. Okay, so we're going to start with just the vanilla flavored adapter at the top. What we have to have are library DNA adapters are going to have sequences which will allow our pieces of library DNA to hybridize to the oligos that are already stuck on the flow cell. So that will be the first step in getting them to become attached, amplified, and sequenced. And we also have to have sequences that will bind sequencing primers that we can use for that purpose later. Okay, so there are other things in there if you're going to have complicated mixtures of samples for indexing purposes to later identify which sample came with which sample. Okay, so we need to have sequences that are complementary to the oligos on the flow cell so that the DNA can bind to it. And we also have to have sequencing primers. Okay, so the additional sequences are basically to specify the sample so that you can have multiple samples at the same time. So we were going to take our libraries with our adapters, denature them, and add them to the flow cell. Okay, what happens next? The DNA will be captured, and this is where we're going to have PCR, multiple PCR reactions going on all at the same time to generate the clusters, which are ultimately going to be the things that we sequence. Okay, so this is from Illumina. It represents what's happening. The actual amplification process, if you do it step by step, is pretty complicated. So we're just going to go with the idea that the oligos on the slide will hybridize to the ends because of the adapters of our sequences, and they will undergo amplification into little clusters on the flow cell. Okay, so the key word here is actually bridge amplification, where the piece of DNA will basically bend over and use the other oligo to which it's hybridized now as a primer to start the next round of PCR reactions. So at the end of this, you end up with pieces of DNA which are actually physically stuck to the flow cell, and those are the ones that are going to ultimately become sequenced. So it does resemble Sanger sequencing, but we have some crucial differences. First of all, we will have 4 color fluorescent nucleotides, and that's fine, attached to with the fluorescent compounds attached to the 3' ends. So those are chain terminators. Okay, and synthesis will be done 1 nucleotide at a time, but occurring at millions of places along. Okay, so of course polymerase will match the coming nucleotide to the DNA template attached to the flow cell. Okay, importantly, these are reversible chain terminators, that they terminate once in order to see what happened in that particular step of the reaction, but then they are removed so that subsequent additions can be made to the same DNA molecule. So this is a crucial difference. Okay, so what's happening in this picture, and it's to illustrate what's happening. Ignore the fact that we're not showing primers, polymerases, or anything. And what we're showing is 2 different molecules that are going to be sequenced in, 5 cycles of chain extension to generate some amount of sequence. Okay, so what we're showing here is the molecule that matches the template, has been identified in solution, it base pairs to it, and it has a color, right? The little green stripes here, etc. Those are the colors of the different nucleotides. So we're going to then start our sequencing, and the first color is going to be as given. So first in this little patch, we have a green and an orange, and those, of course, can be identified as to what nucleotides they are. Right, so that's the first cycle. Right, so each single complementary nucleotide is added, but the reaction doesn't go any further because the fluorescent molecule is attached to the 3' end. So now we're going to excite this with a laser, and that will identify what fluorescence is there at each spot. So the color is going to be determined, and we will capture the image, and the image is recording. Okay, so that's when the reversibility becomes important, because now a new cycle can begin. So the images are going to be captured at each cycle for all the spots on the flow cell. So now we go to cycle 2, right? We've got the same 2 molecules that are being extended, and each one of those acquires the next nucleotide that's complementary to the template. And then the laser will determine what colors those are, assign them to what the nucleotide is, and then reverse the process so that the cycle can begin again. Again, to cycles 3, 4, and 5, each round of extension gives us the addition of 1 more fluorescence image that we will capture one at a time, and then reverse, and then go on to the next cycle. Okay, so here's another representation of the same thing, where we've seen 1 molecule that's being extended over several nucleotides. Okay, and we're showing one at the end with a fluorescence nucleotide on it. And you can follow through what's on the right-hand thing, is an image of that region of the flow cell where the image has been captured over the 6 cycles shown, and you can see which spot this corresponds to according to the color of the spot at that cycle. Okay, yet another representation to envision what's going on. We have a flow cell, and we're showing a teeny little area of the flow cell with a bunch of spots of DNA clusters on it that have been generated by this bridge amplification process. So we're going to look at the molecule on the bottom, on the right, the one that's got a red fluorescence right now, that at the first cycle, it incorporated a C, which was blue, which we can see here, and that's given in the little panel on the right. And the next we had an A, and that was green, and we can see that on the panel on the right. And then finally we have the red. So these images are recorded after each cycle, then the chain terminators are reversed, and the cycle begins again. Okay, so here's a picture of part of an array, and they're telling us that there are 40 million clusters on this particular flow cell. It's kind of astonishing, really. Okay, so analysis. Okay, this is not data you can just sit down and look at, right? There's very elaborate and sophisticated software to deal with the data that is generated, which is, of course, huge if you have millions of sequences that are going on at once, right? So the software is able to remove the extra sequences that come from things like primer, etc., But importantly, the reads need to be aligned to each other and to a reference genome, if you're lucky, right? So there's genome browsers for model organisms that make this very much doable. It's much harder if you don't have a reference genome, but not impossible. Okay, here's a clicker question. I sort of apologize for this one because I made a mistake the first time. Okay, so both traditional and next-gen sequencing use terminator nucleotides that are fluorescent, fluorescent and reversible, reversible. Well, of course, the answer is A, fluorescent. But for some reason, I had the wrong answer in here at first. I will fix this. I had it as fluorescent and reversible, and that's not true. That's just for next-gen sequencing, not for standard sequencing. Okay, next-gen sequencing differs from traditional sequencing in that only next-gen sequencing captures sequencing information in real time, uses DNA polymerase for the sequencing reaction, uses capillary electrophoresis to analyze products, requires primers to start the reaction. Okay, so the answer is A. It's only next-gen captures the information in real time. Okay, so now I want to do one example which has to do with transcription really. It's just a first example of how this sort of technology can help you. And the question we're going to ask is, how do we know where the nucleosomes are in relationship to the DNA sequence? And how can we find things like nucleosome-free regions? Okay, so the question right now is, are these nucleosomes in this particular gene region nicely ordered arrays, or are they randomly distributed? Okay, so here's the idea of nucleosome positioning. We're showing the region of a gene, and we're showing the nucleosomes, and they're arranged as little, we can see them as little peaks where the actual nucleosome is sitting on the DNA. Now, there's such a thing as position nucleosomes where in all the copies of the gene, or most of them at least, the nucleosomes are in relatively the same position. But in other situations, they're not. They're more like randomly distributed. So we're going to consider the idea of nucleosome positioning in this case. All right. So the roles of chromatin remodeling in nucleosome positioning is important. Active genes begin with a nucleosome-free region, or NFR, and that is determined in part by remodeling complexes, but in part also because some DNA sequences themselves do not favor the formation of nucleosomes. And such sequences are often present in promoters in order to assist the process of clearing the DNA of nucleosomes to create an environment that's good or facilitates RNA polymerase binding, etc. So most promoters need help to get the nucleosomes out of the way for the purpose of activating, for the purpose of positioning the +1 nucleotide. Okay, +1, yeah. Okay, so the GRFs are general regulatory factors, and various chromatin remodelers perform these functions. Okay, so here's just a region of a gene is to illustrate the point that some DNA sequences at first don't favor nucleosome positioning there. They tend to repel nucleosomes. But there can be a lot of shifting and sliding around to get the nucleosomes or get the nucleosome-free region where you need for RNA polymerase to effectively bind itself and also for transcription factors to be binding. So there are remodeling complexes that do this, that slide the nucleosomes around, that evict nucleosomes, etc. Okay, so here's our example where a genome-wide method is going to tell us where the nucleosomes are. We're going to use next-generation sequence to do this. Okay, so in this example, we're going to try to relate the DNA sequence to the locations of the nucleosomes. Okay, so in this example, we're going to isolate chromatin and then digest it with an enzyme called micronuclease. Okay, so the nuclease, this is an interesting nuclease because the DNA that's actually physically bound to the nucleosome histone proteins that are physically associated with it are not very sensitive to this nuclease. But the DNA between the nucleosomes and the linkers is sensitive and is digested away by it. So there's going to be a sort of sensitivity region where the DNA is easily digested, and then there's a resistant region, which is those pieces of the DNA, those sequences, which are actually physically attached, or wound around anyway, the histones in the nucleosomes. Okay, so here's an example of a micrococcal nuclease digestion of chromatin. Right, so we're representing the nucleosomes here as being stretched out in a line, and that the region between the individual nucleosomes is more available to the nuclease, with the result that the nuclease eventually cuts the DNA into nucleosome-sized pieces, which is, of course, 146 or 147 nucleotides. So that's the basis of this micrococcal nuclease, sensitivity or not. Okay, so we're going to isolate the DNA that remains after the digestion with the micrococcal nuclease, and all the pieces should be about 147 nucleotides or base pairs. Okay, so the idea here is if the nucleosomes are precisely positioned or phased, all of the cells will have the same piece of DNA associated with each nucleosome. But if the array is irregular, the pieces will be different. The DNA that's protected by the nucleosome, shall I say, will be different according to the location of the individual nucleosome in each cell. So we're going to massively parallel sequence this. We're going to sequencing everything we get, and this would give you the whole genome if you did it this way. We're just looking at one gene. Okay, so we're going to sequence these millions of pieces, and then the sequences themselves are going to be aligned to the genome sequence, so we know where each one came from. So this places the individual sequence according to where it was. Okay, and these alignment is represented as read counts, where each little, there's a little tick mark placed on the x-axis whenever a region is found there in the sequence. And by piling up all these read counts, we can figure out which genes, which places in the genome are more likely to be bound by the nucleosome versus not. Okay, so here's just a representation of what this experiment would look like if we have a regular phased nucleosome array. The nucleosomes are all lined up in a row. And importantly, the exposed region, the linker region, is the same in all copies of the gene, right? The different cells have the same exposed region of DNA and the same protected region of the DNA. Whereas if you have more of a random or an array, but not necessarily phased, we will not have the same pieces of DNA exposed. The same overall amount would be exposed. Each nucleosome still has the same amount of DNA wound around it, but it's going to be different from one cell to the other. So in this example where we have a phased array, if we then do our micrococcal nuclease digestion, isolate individual pieces of DNA and sequence them, we find out that these sequences pile up and make peaks along our x-axis, which is along the gene lengths. And the peaks represent the fact that those pieces of DNA were protected by association with the nucleosome, and the valleys are the regions that are not as represented in the sequence because they were digested away. So the peak tells us where the nucleosome was, and the valley tells us where the linker region was. So this is what you would see for a phased array, where the cells are all basically doing the same thing with their nucleosomes, as opposed to the other situation where you've got as many nucleosomes, but they're kind of scattered along, either in a random order or just in different locations along the gene in the different cells. So this is an example where we can use next-gen sequencing to ask a question about the nucleosomes, but this type of approach is also used for things like what DNA is actually bound by specific proteins, and what DNA is actually bound by RNA polymerase, etc. All right, so I think I just said this. If the nucleosomes are not precisely positioned, the reads will spread out across the gene because the DNA was protected from the nuclease at all locations, not at certain locations. So this kind of same idea is more generally used to locate protein binding regions rather than nucleosome locations. But this I thought was a good example. Okay, so here's a web browser view of the whole thing. Some particular gene in the yeast genome, and the little lumps along the bottom are the positions of the nucleosomes across that gene. Okay, so for example, if we want to know where the RNA polymerase is, where particular transcription factors are, And do these things change when conditions change? Which nucleosomes have acetylated histones, methylated histones, etc. So there are various versions of a technique called ChIP-seq, which we will use to determine these sorts of things. But the idea is to look at the whole genome rather than individual genes. Okay, so we're going to talk about a technique called chromatin IP, which traditionally was just done to look at a particular region, a chosen region of the genome, say a particular promoter, to determine the location of various DNA binding proteins, for example. But this has been brought a few steps further with the addition of ChIP-seq, where you do your chromatin IP and then sequence the results, sequence the outcome across the entire genome rather than just looking at specific genes, etc. So the overall process is more than a little complicated. You have cells that you're going to be using. You then cross-link proteins to the DNA in the cell, lyse these cells with their bound cross-linked protein. And then what you have to have is an antibody that's specific for the protein that you're trying to determine whose location. Now that could be acetylated histones, it could be a transcription factor or whatever else. But you do have to have a specific antibody that's going to bind the protein of interest and eventually pull it down. So having bound the antibody, we're going to then precipitate the antibody complexes with protein A agarose or protein G agarose, which the protein A and G specifically bind antibodies. And so they'll pull down any antibody, well, many types of antibody from the complex. So having cross-linked, shredded our DNA complexes, bound our antibody, precipitate them, we then centrifuge, and then wash, and then we release the reverse, really the cross-link from the antibody, and then sequence the DNA that we get out. So ultimately, we're sequencing the DNA that was associated with the protein of interest. So the antibody was against the protein we're looking for, but we're determining where it was by sequencing the DNA that came down with the protein antibody complexes. Complicated enough? Okay, so this won't work for unknown proteins, but it's a starting point for technologies that can do that. Okay, so this is a protein first approach, and importantly, we need antibodies that specifically bind the protein of interest that we're going to then pull down with the antibody and then determine what DNA sequences throughout the genome where binding occurred. Okay, so I think I just said all this. What I simply did here is just step through the whole process. Okay, so it's important that you precipitate the antigen-antibody complexes along with the cross-linked DNA, and then ultimately sequence the DNA. Okay, so ChIP-seq at the end, when you're looking at this, you will get sequencing data that will tell you what pieces of DNA that you acquired through this process, where they were located in the genome. So here's where you have to get your sequence information and align it to the genome, and you get little peaks where each time the software sees a piece of DNA that matches that, it aligns it, it puts it where it's supposed to be, and ultimately you get a bunch of little tick marks across the genome at the locations where the DNA was pulled down by virtue of binding to that particular antibody. Okay, so in terms of doing this, you really have to have specific antibodies. Okay, so if you're using epitope tagged proteins, that's easiest because you can always buy the antibodies, right? There are actually monoclonal antibodies available to many proteins, not just histone proteins, that can be purchased. Sometimes people have to purify the protein of interest and have antibodies made. But there's an amazing collection of antibodies out there, so that's not usually an important problem if you're working with a model organism. Okay, so here's some sample ChIP-seq data that I pulled from somewhere or another, and what it's doing is assessing the role of two histone acetylases, ESA1 and GCN5, and acetylation of histones H3 and H4, and the relationship of those processes to the binding of RNA polymerase. All right. So this is a complicated experiment. I'm giving this to you as a worksheet. Okay, so we have antibodies. Importantly, we have antibodies to acetylated histone H4 and acetylated acetylated H3 at position lyse 9. And we also have antibodies to RNA polymerase. They don't tell us what subunit. So these were used in separate immune precipitations to pull down the proteins so that the DNA binding locations could be determined. And these were done in wild-type and mutant strains, and these were compared to each other. Okay, so these both encode histone acetylases. So what the results are, we're showing our little tick marks, acetylation for the locations of H3 and H4. Now it's important that those antibodies bind just to the acetylated forms of those histones and not the wild-type or unacetylated forms. Right? It's important that, it's amazing that they can be that specific, but that's important. Okay, so there's many antibodies available.